Okay. Assalamualaikum. Okay. So to prepare for the biochem vivas and lab, uh, the most important thing to begin with is remembering what kind of equipment exists in a lab. So usually it would start with centrifuge machine, thermostatic bath or water bath. We also have shaker mixers. One of the very important and frequently asked machine is the spectrophotometer. We have electric balances, ovens, glucometers, pH meters, electrophoresis machines, incubator, and microscopes available in a lab. Now, centrifugal machine basically works by spinning the given solution at very high rate of speed, and as it is spun at a high speed, the solution separates according to size, shape, density, viscosity of the medium inside the rotor speed. So that is how the centrifugal machine works. Then we have water bath. You can imagine that it's basically a thermostat controlled water temperature system where you can put solutions in if you wanted to have a constant temperature over a long period of time. You did not want any fluctuation in the temperature. You would use water bath for it. Shaker mixer is a very interesting machine. It basically helps by shaking whatever piece of equipment you have. It could be a flask, it could be a tube, a test tube. You basically touch, either you can put it on top of it or just touch it or tap it with the test tube, and it is going to create agitation inside the machine, inside the tube, inside the solution. Electric balance is just like a balance. You can put anything on it, and it'll weigh and tell you, the amount of substance in a material in grams or kilograms or whatever scale is being used. Now come to one of the most important machines. It's called a spectrophotometer. A spectrophotometer can use different types of lights. So, for example, one of the most commonly used one is visible light, UV light, and infrared light. So what does this machine do? You put your unknown mixture or whatever mixture you have in a test tube, stick it inside the machine, and it is going to measure the amount of light being beamed on that test tube, and the amount of light coming out of it. So intensity of the incoming light versus the amount of the intensity of the light that is exiting the substance. So basically, every solution in nature has some absorbance capacity. So that is what the spectrophotometer measures. So the most important use of spectrometry is enzyme assay. So enzyme assay is a biological experiment or biological test that can be performed on a given solution. So enzyme assay would be the primary use of a spectrometer. They can this can be used to identify impurities inside a solution and measure various color components in the visible region of light. So that is how spectrometers work. Now, if you want to be a little more fancy with the concept of photospectrometer, then you should remember that there is this law called Beer-Lambert's law. So, according to that law, the absorbance of a monochromatic light is proportional to the concentration of the sample and the path length, which is like if you are using a test tube, the length of the test tube, the thickness of the test tube, that would measure basically thickness of the sample that you have, that you want to pass the light through. So again, Beer-Lambert's law is the absorbance of monochromatic light is proportional to the concentration of the sample and the path length of the medium. So that is how this machine works. And of course, this is one of the most commonly asked questions. And of course, it can be used to color component in visible region of the light. So color components of the sample. um, Once again, what is the most important use of a spectrometer? Spectrophotometer, it is enzyme assay. So enzyme assay is the most important thing that we do. We also have ovens. You can imagine that ovens are used to heat samples. And then glucometer, there's a possibility that a photo of a glucometer may be shown in the class, excuse me, during the viva, and you would be asked to identify that device. If it is a glucometer, in fact, you could be asked, what two types of commonly used blood glucose level could be. One is called FBS, which is fasting blood sugar level, and then RBS, the random blood sugar level. So those are the two measuring levels, two glucose measuring levels that are commonly used. Now, what is the normal fasting blood sugar level? That answer should be 100, although it should be a range, but in our understanding, based on the content of the class, the teachers want to hear that fasting blood sugar level is below 100. The other instrument in the lab available is the pH meter. I am not going to. Yes, less than 100. No. Random could be anything, if you're measuring depending on when you measured it, whether you measured it after food, immediately after food, or in the morning, or in the afternoon, that could vary. But fasting blood sugar should be 100. So fasting would mean when you have not eaten for about 8 hours. Um, uh, Again, going forward, pH meter would be to measure the hydrogen ion activity in a water-based solution, which would indicate whether a solution is acidic or alkaline. Moving on to the next very important device is called electrophoresis. Electrophoresis is a device where you can put a sample in that device, and it is going to create an electric field around that sample. So if you have charged molecules, uh, they would be separated according to the electric field. How is it used in the lab? It is commonly used to separate DNA and RNA or protein molecules based on their size and their electrical charge. So electrophoresis is used to separate DNA, RNA, or protein molecules based on their size and electrical charges. So that should do it. And of course, we have an incubator. Incubator basically provide a controlled environment for a specific temperature on a long-term basis. Could be several hours to several days and weeks. They can help, when we need microorganisms to grow in a certain temperature. That is what we use. Okay, let's move on to one of the labs that we had done in the class. That initial lab that we did was dealing with different solutions. The definition of a solute, that it's a substance that dissolves in a solvent. A solvent is a substance that has the ability to disperse a solute. That's called a solvent. A solution is a homogeneous mixture of a single phase or a single phase mixture of a molecule, atoms, or ions of two or more components. That's called a solution. Um, there is a concept called emulsion. Uh, Ahmad, you wanna give it a try? What is an emulsion? Give an example of an emulsion. Okay. Hmm. Yeah. So if Mhm. Go ahead. Go ahead. Hmm. Right. So, yeah, right. So emulsion, there are two concepts that are important when it comes to emulsions. Emulsion can be temporary and incomplete, or it could be permanent and complete. So, how do you create a temporary and incomplete emulsion? You put a little bit of oil in water and shake it vigorously. That oil is going to get dispersed in small droplets throughout the water solution, and as long as you keep it shaking and agitating, you will see cloudy appearance. Cloudy appearance, and that cloudy appearance is called emulsion. Now, if you stop shaking it, stop agitating it in a moment or two, the two layers would become visible. The oil would separate, and the reason was that this was an incomplete and temporary suspension, excuse me, temporary emulsion. If you wanted it to be permanent, in that case, you are going to need some type of emulsifying agent. And one of the emulsifying agents is bile salt. So if you have water and oil and you want to emulsify it on a permanent and a permanent basis and completely, then you would add bile salts in it, and that would give you a permanent cloudy shape or structure inside the test tube. Now, what is the significance of emulsions? Emulsions basically increase the surface area of the oils so that enzymes can work on them. So when we eat lipids or fats, they cannot be worked upon if they are in clumps or in big droplets, big drops. So they have to be emulsified. They have to be turned into those that cloudy form. So very little tiny molecules could be made so that enzymes can work on them. They can break them down. They can, they can be small enough to be absorbed in the microvilli. There's another concept called suspension. A suspension is a solution in which a particle size of the solute is greater than 200 millimicron. So when suspended, this solution is called a suspension. Now, this is an extremely important concept in pharmacology because we have a lot of drugs that are sold in a suspension form. Why? Because the drug may have a very disagreeable taste. Yeah. So the suspension could make it, you could put different types of taste agents, cherry or lemon or whatever, taste you want. So that makes it more palatable. Becomes ideal dosage technique for patients who have difficulty swallowing tablets. In the case of children, elderly patients. So and also it is very easy to absorb from suspension than tablets and capsule. It's faster. um, so that's suspension. Now, emulsions are also used in the case of medicine. So sometimes we may need to mask the taste and smell of an oil. So in that case, emulsion may be important, and oil becomes easily digestible. And of course, it also leads to uniform distribution of the drug within the emulsion. So I think we can stop here, and then we can have another video recorded for the specific schemes for testing carbohydrates, proteins, and the lipid tests. Okay, thank you. Assalamu alaikum. Oh, you're welcome. Okay. Okay. Um, yeah, yeah, yeah. Absolutely. Absolutely. We should. We absolutely should. We absolutely should. Take it. So, did you stop recording?